Our next speaker is Professor Ashley Bush from Australia. I should say very good morning, Dr. Bush, and thanks for joining us. Dr. Ashley Bush is a psychiatrist and translational neuroscientist. He is the director of the Melbourne Dementia Research Centre and head of the Oxidation Biology Unit at the Florey Institute of Neuroscience and Mental Health at the University of Melbourne. Professor Bush discovered brain metal metabolism as a major factor in Alzheimer's disease pathogenesis and focuses on the neurobiology of metal ions and oxidative stress in neurodegenerative diseases and psychiatric disorders. Today he's going to talk to us about iron, ferroptosis, and AD. Dr. Bush.
Yes, can you hear me?
Yeah, we can hear you.
Okay. Well, thanks very much for the invitation to speak here today, and thanks, Rams, for organizing this great seminar. I'm going to be talking today about iron in particular and its role in Alzheimer's disease, and some of our recent work in terms of parsing the contribution of iron to the pathogenesis of Alzheimer's disease. And of course, Alzheimer's disease is known as a proteinopathy, but it is also a metalopathy. Certainly if you examine the classic proteinopathies of Alzheimer's disease, you see high condensations of transition metals. And in particular, I'm featuring iron here in the amyloid plaques and condensing within the neurofibrillary tangles. And there's also obviously changes in the background tissue, which I'm going to be elaborating on right now. Interestingly, the accumulation of iron within the tissue has been described as a feature of Alzheimer's disease pathology for quite some time. And here's the first instances in the literature that I could find from 1953 and 1960 that comments on the large amount of iron staining that's occurring within the tissue in Alzheimer's disease. If you fast forward to the modern era, I was commenting on the pathology of Alzheimer's disease being characterized by a large amount of iron accumulation. And this shows you more modern images from Louise van der Weert's lab that show the histology of iron accumulation within the neocortical areas in Alzheimer's disease. And so it compares the normal stain. This is using a modified Pearl stain of the on the left of a normal piece of cortex, and compared to the middle and the right-hand sides, which shows the increasing amount of iron which becomes stainable within the cortical lamina and within the amyloid plaques themselves. And then on the lower part of the slide, you see the iron accumulating in both neurons and in activated glia. And so this iron had been known for quite some time, but had been rather underappreciated. But we've taken a renewed interest in this, particularly over the last 5 years. I've been collaborating with the Rush University Medical Center, Martha Claire Morris, who sadly passed away during this study, as well as Julie Schneider, in examining large cohorts of tissue by ICP mass spec. And so this is an analysis of 680 post-mortem human brain specimens that come from this cohort where there is antecedent cognitive data for up to 10 years prior to death. And we're going to analyze this tissue for its metal content and see how it relates to the pre-mortem state of the patients. So what I'm going to show you today is just an analysis of the 227 brain specimens that we have that were positive both for a diagnosis of Alzheimer's disease as well as for amyloid pathology by CIRAD neuropathology criteria. And this is a summation of examination of 4 different brain regions in these 227 cases. So to summarize a lot of data, and I'd certainly encourage you to have a look at these papers which we've recently published on this, I'm just going to start off by showing the contributions of the various pathologies. Firstly, neurofibrillary tangles, we're using this as a reference point since it's well accepted that the neurofibrillary tangles are associated with longitudinal cognitive deterioration. On the x-axis is years prior to death, and on the y-axis is change in global cognition based upon the reports of their antecedent cognitive performance. And you can see that if you organize the data in terms of tertiles of neurofibrillary tangle burden, that people with the highest burden of neurofibrillary tangles deteriorated fastest prior to their death. So this is a reference point. This is already well accepted in the literature. So now if you have a look at iron in this tissue, we can see something similar, and the data are adjusted for the contribution of the other pathologies. So iron as an independent variable also was concordant with the antecedent cognitive decline. So the tissue that had the highest iron levels was coming from subjects who had had the most aggressive cognitive decline prior to their death. And then finally, if you look at the amyloid contribution, it's rather disappointing because there's no impact of amyloid burden on the antecedent cognitive decline. This would suggest that iron represents an independent factor in terms of neuropathology that may contribute to the rate of cognitive decline in Alzheimer's disease. And this signature of iron burden accelerating cognitive deterioration is seen in other means by biomarkers such as CSF ferritin levels and neuroimaging. Here's the CSF ferritin levels, which we published recently, that shows that those with the highest levels of CSF ferritin have got a far more aggressive form of deterioration than those with lower ferritin levels. And finally, we've done neuroimaging using quantitative susceptibility mapping, which is a form of MRI that can sense the burden of iron within the brain. On a cross-sectional basis, you see a small elevation of iron within the neocortical structures in Alzheimer's disease. But on the left, you see a graph which shows the change in Z-score in terms of cognitive performance over a period of 6 years. And this is a prospective study done with the ABLE cohort. And on the x-axis of the graph is the QSN values, which represent the iron burden of the tissue. And this is in the hippocampus. In subjects that did not have amyloid detected in their brain by amyloid PET scan, the relationship of hippocampal iron to the cognitive performance over the 6 years was negligible. So iron did not impact upon the rate of change in cognitive decline. However, in subjects who had amyloid within their brain, who were amyloid positive, there was a very strong interaction. So in people who have got an amyloid pathology, which can of course be asymptomatic, and it's asymptomatic in the preclinical phase of Alzheimer's disease for up to about 15 years on average, those individuals are very vulnerable to elevated iron, so that the higher iron levels are associated with a much more aggressive cognitive decline over a period of 6 years. So iron is certainly featuring in Alzheimer's disease pathology, and the question has been, where does it actually feature? Now, our thinking, and I thank previous speakers for mentioning this, but we had also thought that perhaps iron was interacting with soluble beta amyloid and forming plaque, or it was interacting with soluble species of tau and promoting neurofibrillary tangle formation. All that might still be true, but from our data from the post-mortem studies, we can see that even when we subtract out the contributions of plaques and tangles, there's still the burden of background tissue iron seems to matter. And so we've changed our thinking a bit about how iron fits into the pathology of Alzheimer's disease by putting it more downstream, so that even in tissue where there is plaque and neurofibrillary tangles, the role of the tissue iron in the background promotes neurodegeneration. And after all, it is the death of neurons which is the most important event in terms of causing dementia. And so we hypothesized that this burden of iron is promoting neurodegeneration, and a candidate mechanism for this is ferrotosis, and I'll explain this as we go along. So one of the major questions that we wanted to answer is, could there be could there be an explanation for the chemical changes in the brain that occur with aging that would increase the risk for neurodegeneration? And could that factor be iron? And indeed, the literature has filled up with instances of iron accumulation within the brain with aging. Here's a couple of papers. One of them is from 1958, which surveyed the levels of brain iron throughout the human lifespan. And levels of iron within the brain are quite low at birth, but then they rise very quickly in childhood, and they begin to level off in early adulthood, but they continue to creep up throughout the human lifespan. And the image below that, I show a reference, much more recent reference of this same phenomenon for one part of the brain, which has used R2 star measurement of MRI images to quantify the iron burden within the putamen. You see the same phenomenon. That's very low in childhood and it rises steadily with aging. It plateaus in middle adulthood, but it continues to rise slowly. And indeed, you see this age-dependent elevation of brain iron in rodents. Here's one of the first papers we had on this subject, where we measured the iron levels within the brains of mice as they aged from 2.8 months to 8 months to 18 months. In the normal wild-type mice, you get this increase in brain iron. In the APP transgenics, which is the model for Alzheimer's disease, curiously, they still have an elevation of iron with aging, but they have an effect of the transgene lowering the levels of iron within the brain tissue. So this is the first clue that APP has an impact upon iron homeostasis. And we thoroughly characterized this iron elevation in the brain in rodents and showed that there are both sex and genetic background impacts on the elevation of iron. And more recently, we've shown that APP knockout mice have got an exaggeration of this elevation of iron that occurs with aging within the brain. And I'll explain about APP's role in iron in a minute. So the literature subsequently filled up with many instances that reported an elevation of iron within the brain as a consequence of aging. So I've cherry-picked a few papers here. This has been reproduced in rodents, it's been reproduced in primates, and it's been reproduced in humans, where the work is capitalizing on the use of MRI to quantify the iron burden within the brain with aging. So now that there's quite a robust literature about this iron elevation accumulation within the brain with aging. So the elevation of iron can cause problems through a number of means. I'm sure most of you are familiar with Fenton chemistry, where iron can form the harmful hydroxyl radical, which can then react with any target that it's adjacent to. But an elevation of iron can also impair antioxidant systems, and here is one instance where HIF1 alpha can be degraded. And I'm going to focus a bit more now on ferrotosis, which is an iron-dependent form of programmed cell death, which was described in 2012, and it involves the combustion of iron with long-chain fatty acids to generate lipid peroxides. And here's a very nice illustration of oxidized arachidonic acid within an amyloid plaque. And the literature is very clear that lipid peroxide species are markedly elevated within the brain in Alzheimer's disease. And so this is indirect evidence of the presence of ferrotosis. So what is ferrotosis? Ferrotosis is a reaction that leads to cell death of the products of ferrous iron in the cytoplasm with polyunsaturated fatty acids. And the lipid peroxides that are formed can lead to disruption of the plasma membrane and cause cell death. This reaction can be blocked by small molecules, and vitamin E is a very safe but very low effectiveness molecule in terms of blocking the lipid peroxide. It can also obviously be blocked by chelators such as deferoprone, which is in clinical trials. But I think what's relevant for this particular conference is that the lipid peroxides, when they break down, create reactive aldehydes such as 4-hydroxynonenal and malondialdehyde and other species. And these reactive aldehydes like to react with proteins and cause adduction and protein cross-linking. And I'm not showing any of the evidence for this today, but it's in the literature that this reaction can contribute to the formation of amyloid plaques, neurofibrillary tangles, and other neuropathologies such as Lewy bodies. So I believe that this lipid peroxide reaction is one of the most critical events that facilitates the mixture of features that you see in Alzheimer's disease neuropathology. Another feature of Alzheimer's disease neuropathology is a drop in brain glutathione levels, and glutathione plays a critical role in quenching the lipid peroxides that are generated during this reaction through the activity of an enzyme GPX4, glutathione peroxidase 4. So why is the brain so vulnerable to ferrotosis? Well, it has the following risk factors. It's got high and dynamic iron concentrations. It's postmitotic tissue. It's got a high oxygen turnover. It's got a high content of polyunsaturated fatty acids. And as I've emphasized before, it's got this age-dependent increase in tissue iron, so that the iron accumulation will eventually cause some stress upon the systems that prevent the lipid peroxidation from going out of control. In Alzheimer's disease, you see changes within the tissue that are consistent with ferrotosis. And I'm just listing them all here. These type of features are seen in other neurodegenerative disorders, and they affect different nuclei, obviously. So I've argued that this iron could be causal in terms of neurodegeneration, but really the test of the argument would have to be whether the genes that are implicated in Alzheimer's disease and other forms of dementia are involved with ferrotosis or iron metabolism. And I'm not going to have time today to go through all of our evidence for this, but this is where the campaign of the lab currently exists. And I'm going to present to you the data about the amyloid protein precursor, but we're looking at presenilins and apolipoprotein E and tau in iron homeostasis and in ferrotosis, and I can tell you that they're all participating. But for today's presentation, I'm just going to focus on APP to show you the evidence for APP's role in iron homeostasis. Now, we have been publishing over the last few years on the physiology of APP in iron homeostasis. Iron is effluxed from all cells through ferroportin. It's the only channel out for iron, and it gets loaded onto transferrin and transported away. And what we've picked up on is, starting off with Jack Rogers' paper, that there is an iron responsive element in the 5' UTR of the APP transcript. That iron induces APP production on a translational level. And tau, the other protein that forms neurofibrillary tangles, has a role as a microtubule-associated protein to facilitate the trafficking of APP towards the surface under high iron conditions. And then the APP is presented to the surface where it binds to ferroportin and stabilizes the ferroportin, allowing iron efflux to be promoted. And so we've published quite a bit on this over the last few years, and it's been replicated by other groups. I'll just show you a little bit of the data about how the amyloid protein precursor behaves biochemically. You can co-immunoprecipitate ferroportin with the amyloid protein precursor out of both human and mouse brains. So it's a major interaction, and when we do bookkeeping on it, we find that 70% of the amyloid protein precursor in the brain co-immunoprecipitates with ferroportin. And if you look at the amyloid protein precursor knockout neurons in cells in culture and load them up with radioactive iron, the efflux of iron is much slower from the APP knockouts compared to wild-type cells. So it has a really marked impact on iron homeostasis. Now, our most recent work on this is to have a look at mutations of the amyloid protein precursor that are known to affect its processing. The amyloid protein precursor is cut into the A beta fragment through the action of a beta secretase called BASE at the minor terminus of the A beta, and by carboxyl terminal cleavage through the gamma secretase. And the alpha secretase plays an important role in preventing A beta generation by cleavage at residue 16. Now I'm going to show you the data from studying two mutations of the amyloid protein precursor at residue 2 of the A beta sequence, which is highlighted in yellow here. Mutations to valine cause it's called the Italian mutation, and this is an aggressive form of familial Alzheimer's disease with exaggerated pathology and an early onset of dementia. Mutation to threonine is protective. This is called the Icelandic mutation, and people who have inherited this mutation are actually protected against Alzheimer's disease and have got less pathology as they age. Now, these mutations have been used to argue that the beta amyloid generation is causative in Alzheimer's disease. But of course, as I'm sure you're well aware, it's been frustrating to try to generate curative interventions on the basis of clearing amyloid. And one of the questions we asked is, well, what impact could these mutations have on iron homeostasis, and could that be an alternative explanation for the neuropathology and the neurodegeneration that occurs in Alzheimer's disease? So we studied the effect of these mutations on cells in culture, and specifically on this iron homeostatic pathway that we had been elaborating. So just show you a few images from this paper. We took N2A cells that had been challenged with iron, and you can see that the effect of iron is to drive up the concentration, and then modulated the processing either with an alpha secretase inhibitor, which is TAPI2, or a beta secretase inhibitor, which is B4. You can see the alpha secretase promotes the retention of iron, and beta secretase inhibitor prevents the accumulation of iron. And this is accompanied with the position of APP on flow cytometry being increased on the surface of the cell. So the effect of the B4 beta secretase inhibition is to promote both APP localization on the surface of the cell and co-localizing with ferroportin. So that's how we believe that it acts to prevent the accumulation of iron within the cell. Now, if you study the same experiment, this time by using RNAi against the alpha secretase itself, you cause an accumulation of iron within the cell. And if you knock out base with RNAi, you get you normalize the amount of iron retained within the cell, and you get the commensurate localization of both APP and ferroportin. So the take-home message here is the alpha secretase and beta secretase cuts both influence the ability of the cell to retain iron, and so therefore we tested what the impact of that is with the actual pathogenic mutations. So here you see the accumulation of iron in cells that have been challenged with iron. You can see that the accumulation of iron is, as I mentioned before, blocked by a beta secretase inhibitor. The Icelandic mutation does not accumulate much iron at all when challenged by iron, and beta secretase inhibition normalizes what little bit there is there. But curiously, the Italian mutation, which is the pathogenic mutation, retains a great deal more iron when these cells are challenged with iron. And once more, this is normalized by blocking the beta secretase activity with the B4 inhibitor. And as we expect, the location of the amyloid protein precursor matters so that the Italian mutation has less APP on the cell surface, and the Icelandic mutation has more APP on the cell surface compared to wild type. And if you look at ferroportin, there is a commensurate co-location of ferroportin on the cell surface. So the protective Icelandic mutation appears to protect against iron accumulation by allowing more APP to be stabilizing ferroportin on the cell surface. And this is the first instance where pathogenic and protective mutations that are related to the risk of Alzheimer's disease have been studied in terms of iron metabolism. And this is the beginning of the journey, and we hope over the next while to be able to elaborate this. Just to summarize, what we think is happening is that normally soluble SAPP alpha or full-length APP will stabilize ferroportin and facilitate iron efflux. But under conditions that promote beta secretase cleavage, the SAPP beta sequence cannot stabilize ferroportin, and the ferroportin becomes internalized and degraded and does not allow the efflux of iron, which builds up within the cell. So just to finish off, these findings inform decisions about clinical management. We expect them should be tested in clinical trials. There was actually a phase 2 clinical trial of desferrioxamine, a well-known iron chelator, which was published in Lancet in 1991, and actually reported a benefit in terms of outcome in Alzheimer's disease. More recently, one of the few successful phase 3 clinical trials of Alzheimer's disease reported the effects of vitamin E, which is an anti-ferrotosis agent and has been shown to be effective in this phase 3 clinical trial in mitigating the symptoms of Alzheimer's disease and the long-term outcome. Finally, most recently, we're expecting results from a phase 3 clinical trial in Parkinson's disease of an agent called deferoprone, which in phase 2 did seem to modify the disease. And in Parkinson's disease, there is the same problem of iron elevation that occurs within the tissue. And so this was a proof of concept, which we're also checking out in a phase 2 clinical trial in Alzheimer's disease. And hopefully we'll have results on that over the next 12 months. So thanks for your patience with my electronic problems. I just credit the team back in Melbourne, a large number of collaborators who we have, and I've featured mostly the work of Scott Aiton today, but also the collaboration we have with the Rush University Medical Center and Lund University Medical Center with Oscar Hansen. So credit the support from these agencies, and thank you very much for your attention.
Thank you very much for the lovely talk. We have questions in the Q&A, the chat box. Let me read it for you. Nicholas Filler asking, why do you propose iron accumulates so much more in older individuals than younger ones if it is not directly linked to amyloids?
Sorry, could you just say that?
Okay,
again?
why do you propose iron accumulates so much more in older individuals than younger ones if it is not directly linked to amyloids?
Well, the iron will accumulate inevitably, so it accumulates in everyone, and it's the data that we have here indicates that there might be an association between the accumulation of iron and the formation of pathology, but we could see more from the data that we got in the post-mortem tissue that that iron elevation is working independently of the pathology, because we could account for cognitive deterioration separately when you looked at the contribution of tangles versus plaques versus the background iron levels. So the elevation of iron is a feature of aging itself, and it may also contribute to amyloid formation and tangle formation. We did see an interaction between iron and tangles, so it might support the concept that tangle formation is a consequence or associated with iron accumulation. But that part has not been proven from the data that I've shown you. There's other animal data that shows that, for example, the tauopathy model of Alzheimer's disease, that you do get iron accumulation as a consequence. We think that if you damage tau solubility by forming neurofibrillary tangles with a genetic strategy, that you will get iron accumulation as a consequence of not being able to express enough APP on the surface of neurons. So these are pretty complex models, but I think bit by bit we're sorting this out.
So another question from Feng Ding. Could iron accumulation be the consequence of amyloid aggregation, serving as the reservoir for iron?
Well, we checked on that, and it didn't seem to be, but the amount of iron that's trapped in the amyloid is probably not going to be picked up when you measure the iron on the basis of whole tissue. I think there's certainly iron in amyloid, but the volume of amyloid might be less than 1% of the contribution of iron when you measure a piece of tissue. So although amyloid is very vivid and you can see it very clearly histologically, the quantity of iron that's present in there is only a small fraction of the total iron of the tissue. So I think it's unlikely that the iron elevation is due to either proteinopathy trapping iron. I think it's got to do with the background tissue levels rising in all the cells.
So you mentioned about lipid peroxides playing a role in the iron accumulation and the pathology of AD. So what's known about the lipid peroxides for other metals? Do they promote and play a role in the pathology of AD and other neurodiseases, like zinc and copper?
Well, yeah, so zinc is not redox active. So it itself is not going to participate directly in catalytic lipid peroxidation. And copper is redox active, but its abundance is much lower than iron. And free copper is available at much lower concentrations in the cytoplasm of the cell compared to free iron. So the concentration of the labile iron pool is estimated at about 2 micromolar. The concentration of labile copper is estimated in the femtomolar range. So it's just simply not as available. So that for that reason alone, I think iron is probably the most important candidate in terms of generating lipid peroxidation in any system.
Great. I don't see any other questions. Thank you very much for the nice talk and the Q&A session. And I would like to thank all the speakers for the great talks and sharing your great findings in this important area of research. I also would like to thank the chairs and the participants. I'll see you all in the next meeting. And we've been zooming in since morning. It's quite late evening. Yeah. Thank you very much.
Yeah. Okay. Thanks very much.